Hi everyone, welcome back to my YouTube channel. My name is Rosie. If you are new here, please do subscribe. So today I want to talk to you about my genetic consultation that I had with King's Fertility because me and my husband are doing embryo freezing. Now he is in the other room because he didn't want to be filming today, which is absolutely fine. This is my YouTube channel. But I kind of wanted to talk you through everything that we talked about during our consultation. I do have a list of notes here. So basically I recorded the conversation just for kind of my own personal use so that I didn't forget any information. I've gone through and listened to the whole thing and taken notes. So I'm going to go through my notes and talk to you guys about what we spoke about during the consultation. Now just to give you a little bit of backstory, I think this is episode 6 now. I might be wrong, but I think it's episode 6. So there's 5 other episodes talking about our journey so far, talking about my history with 3 miscarriages and an abortion. But basically my husband and I are both carriers of a very rare genetic disorder called Ellis-Van Creveld. In 2017 we were pregnant and found out that our baby was a carrier, no not a carrier, had Ellis-Van Creveld. So we had to have an abortion at 26 weeks, so I was 6.5 months pregnant, had an abortion, and it was confirmed after the autopsy that baby had Ellis-Van Creveld and that me and my husband are carriers. So since we found that out, we tried getting pregnant 2 more times, had 2 miscarriages, and we don't want to have children now, but maybe in the future. So we want to do embryo freezing. So I'm just kind of giving you a little bit of information to make this video make sense. So the reason we spoke to the genetics team at King's Fertility is because we're going with apricity for our embryo freezing, but King's Fertility for the testing part of it. Now most people who freeze eggs or embryos or do IVF don't need to do this section at all, but if you do know that you're a carrier of a certain gene, you and your partner, and you want to make sure that the embryos that you implant do not have this gene, this is what I'm going to be talking about today. So basically we said hi, introduced ourselves, we went over some basics like our name and date of birth. We talked about my previous experience with miscarriages and abortion, and also finding out about Ellis-Van Creveld, how that happened. So that was kind of just to give her an insight as to our past history, because like I said, most people don't do PGT-M testing, which is what we're doing. So I think she kind of wanted to know why we were looking into it and what we kind of knew so far, I guess. So from there, we talked about EVC, which is Ellis-Van Creveld, and something she told us was actually that when we do the EVC testing, so Ellis-Van Creveld testing, they will also test the chromosomes. So in my previous video, me and my husband talked about our initial doctor's appointment, and she suggested that because I'd had 3 miscarriages that I should probably do chromosome testing to make sure that the embryos don't have any chromosome abnormalities, because that's the main reason that people miscarry, is that the baby has a chromosome problem. So she talked about the fact that when we test for EVC, they kind of find all of the information about everything, so including the gender of the baby, including any chromosome abnormalities. So we'll have all of that information, so it actually isn't an added cost. Because in the last video we talked about the fact that we thought it was going to be an extra cost of about £500 per embryo, which we were then like, oh my god, it's more testing, more money, more time, more this, more that, but actually it's not anything more, it's just kind of all included with the PGT-M testing. So that was really brilliant that we kind of knew that. And she mentioned that we will see everything and we will receive 2 sets of information. So she was talking to us about the fact that there's kind of 4 possibilities. So either the baby has Ellis-Van Creveld, or the baby is a carrier of Ellis-Van Creveld, or the baby is not a carrier of Ellis-Van Creveld, and also chromosomally normal and okay, or chromosomally abnormal and not okay. So obviously you can have a mix of the 2 there. You can have a baby that doesn't have Ellis-Van Creveld but does have a chromosome abnormality. You can have a baby that does not have a chromosome abnormality but does have Ellis-Van Creveld, or you can get babies that are like not carriers and not chromosomally okay. So there's lots of kind of different variations of this, but basically in order to implant we need a baby that is chromosomally okay and either a carrier of Ellis-Van Creveld or not affected by Ellis-Van Creveld. So we can't implant a baby that has a chromosome abnormality or Ellis-Van Creveld. I hope this is all making sense. If not, make sure you let me know in the comment section down below. I'm happy to answer any questions that you might be having. So she talked about the fact that the lab has confirmed that they can test for Ellis-Van Creveld and it's approved by the HFEA. Now the HFEA is the governing body in the UK that oversees all kind of IVF treatment and special cases, I guess. So that sort of shows that Ellis Van Creveld has been tested for before. And in order to get approved for PGT-M testing, it has to be quite a severe syndrome, like bad enough for them to confirm that it would have life-altering effects on the baby or something like that. I'll try and find the definition, but it's quite, you know, they can't just test for everything. So it is quite a serious syndrome, Ellis Van Creveld. But it is good that it's already approved by the HFEA, because otherwise we would have had to apply for a licence and get that approved, and it might have taken, I don't know how many months longer, but it's good that that's kind of already been done. Next, we talked about our family history. So we filled out a questionnaire talking about, okay, mother, are they still alive, yes or no, date of birth, date of death if they're not still alive, and then cause of death. And we did that with parents, brothers, grandparents, aunties and uncles, cousins, and like anyone else you could think of, so close family. And we kind of went through, and she was just kind of confirming all the information basically. And there's been a couple of people in my family that have had cancer. So she wanted to know about the age of diagnosis, because sometimes if you get diagnosed, you know, with lung cancer at age of 80 after smoking 50 a day for the past 80 years, it's kind of because of the cigarettes. Whereas if somebody gets diagnosed with cancer at an earlier age, that can sometimes, I don't know, I don't want to say something I don't know for sure, but she said that if it's an earlier diagnosis of cancer, that's something that she would want to know about. So I had to then go and ask my parents about the cancer history in my family, about what ages people got diagnosed with. And we also talked about my dad's mitochondrial disease, but that's passed through the women of the family. So my dad can't have passed it on to us because he's obviously a male. But we had to kind of go through all the family history and talk about anyone who might also be affected, or anybody with the Ellis Van Creveld, or anybody who might have different things that we need to keep an eye on, but it all seemed okay. So again, we talked about why we're wanting to freeze our embryos. So I kind of mentioned that we don't want kids now, we want to travel, but as I'm 34, I want to be able to freeze my embryos to maybe use it in the future. And we've kind of talked about it, that if we ever want to get pregnant in the future anyway, I think we would do IVF because of the Ellis Van Creveld, and try and eliminate that so that I don't have to have an abortion. And if we're doing IVF anyway, it kind of makes sense to do the IVF section, as in like the embryo freezing now while I'm younger, and then hopefully we'll at least have a couple of embryos to play with when we're older. So that was kind of part of the conversation there. And also Jeremy mentioned about kind of using technology to reduce miscarriage and abortion and make sure any future pregnancies are easier. Because obviously if we got pregnant now naturally, we might have a baby with Ellis Van Creveld, which would mean another abortion. We also might have a miscarriage. And she said that a lot of miscarriages happen when there's a chromosome abnormality. So it's not because of the Ellis Van Creveld, it would have been a chromosomal problem. So if you can have an embryo that doesn't have Ellis Van Creveld and doesn't have any chromosome abnormalities, you're more likely to have a healthy pregnancy. So that's kind of also part of the reason we're doing this. If we ever want kids, we kind of want to give ourselves the best chance of not having miscarriages and not having abortions, because it's not fun to go through. So that was talked about. She asked us about how we're practicing self-care in terms of doing yoga or meditation and things like that. And I also mentioned that I had been going to therapy and I didn't like them, so I wanted to look at someone else. And so my last video, I actually did a video talking about BetterHelp and my kind of overall opinion. For me, it wasn't for me. So I sort of said that to her, and she said that King's Fertility do have therapists I can talk to if I want to get in contact with them. I haven't done yet. I feel okay at the moment. I don't feel like I need to talk to anybody at the moment, but yeah, that is an option, which is good to know. So we then talked about testing in France, because we had the abortion in France and we had blood tests to confirm that we are both carriers as well. And we did see genetic doctors when we were there. Obviously it was all in French, so I didn't understand a lot of it, and we were heartbroken and going through like a massive trauma, so I don't think I wasn't particularly paying attention. But I think she was just kind of getting an idea of how much we know about Ellis Van Creveld and about chromosome abnormalities and that kind of thing. And then she went on to talk to us about what are cells, what are chromosomes, what is genetic information. And she was actually really, really good, because even though she dumbed it down to like kid level so we could understand, because obviously we're not doctors, it didn't sound patronising or like we were stupid. So I think she was really great at kind of explaining the situation, explaining what the testing does and why they do it without making us feel stupid, which I think was really good. So she talked to us about what is Ellis-Van Creveld and the features of the syndrome, which got me a little bit upset. She actually had a whole presentation. It was really good. She had a presentation showing like what cells are and what chromosomes are and how you test the chromosomes. And she had a slide about Ellis-Van Creveld talking about the different features. And I got a bit upset because I was like, yep, our baby had like all of these things wrong with him. Like he had like, I think we counted once, maybe like 20 different things wrong with him. So it was not even just like one or two, it was like everything wrong with him. So she'd obviously done some research, but she said that there's 20,000 genes and people can carry lots of different conditions. And even though she didn't know about Ellis-Van Creveld beforehand, she had done the research to kind of find out any information that might be useful for her. So as we were talking about this, we talked about the fact that there's a 25% chance of having an affected baby, a 25% chance of having an unaffected baby, and then 50% chance that the baby would be a carrier. And that's something to question, is if we would implant a carrier baby or not. So I think we would go for the non-affected first and then the carriers after that. I mean, we have no idea how many we're going to get at the end of all this, so it's kind of hard to say. But I think she was just kind of explaining all of that to us, which was useful. We then talked about PGTM and what that means. So PGTM is when you look for a specific gene defect before implantation. I think it's monogenic or monogenic or something. I'm going to leave it on screen what PGT-M testing means. But basically, like the chromosome abnormalities test for things like Down syndrome and the other syndromes. I can't remember, she said them, but I can't remember them now. Whereas PGT-M will look specifically for Ellis-Van Creveld syndrome. It won't look for any other syndrome, just Ellis-Van Creveld, because we both know we're carriers. So that's really interesting. So even though we test for Ellis-Van Creveld, we could have a baby that has a different syndrome because we're not testing for that. And she kind of talked about, like I said, about the chromosomes, and she was saying, for example, if she actually used a pen as an example, and she said, for example, if there's lots of different coloured bands along the pen and you have another pen here, you want to see if this green band is the same as this green band or if this one is blue instead, and that would be the gene defect. So it was really interesting the way that she kind of explained it to us and made us feel like we understood what was going on. So the next thing she said was that with PGT-M testing, they usually use ICSI. I'm going to leave that on screen. It's basically where they take one sperm and insert it into the egg. And she said that, let me just read my notes here. So it can reduce contamination. So sperm released near egg cells, there's other sperms that can stick to the outside of the egg cell and contaminate the egg. And also there's granulose cells, I think that's what she said. And with ICSI, they would strip all the cells from around the egg. So there's only one sperm injected into the egg, and it really reduces the contamination and any kind of problems that you might have. So if you were to have sex naturally, or if you do IVF without ICSI, you kind of have the egg and you pop a load of sperm in there and the sperm just goes at it, and it's the first one that enters wins, basically. But like I just read out, if another sperm attaches onto the outside, that could contaminate the pregnancy, or also the cells around the outside could, what did I say about that? Granulosa cells. If they get them cleaned up, it can just reduce any kind of problems, basically. So we're going to go ahead and do ICSI. We didn't really know what it was, but it seems like the best option for us. We've been told that a couple of times now, so we're going to listen to the doctors. I kind of thought that ICSI was only for people who couldn't get pregnant, like if their sperm wasn't as good, but Jeremy's sperm is fine, and obviously we've been pregnant 4 times, so we get pregnant quite easily. But actually it's not to do with just how easily you get pregnant, it's also to do with the quality of the embryo afterwards. So quite happy to move ahead and do that. So she mentioned that embryos grow to day 5 or 6, so you know they multiply every day. They start off as 2 cells, well probably 1 cell, and then 2 cells, 4 cells, 8 cells, whatever. And on day 4 or 5, they take a sample from the placenta to test so the baby is not affected. I think in the video where I talked about the doctor's appointment, I talked about it as if it was that appointment. It wasn't, it was this appointment. And I think I might have said the amniotic fluid, or I said something different, but it's the placenta. So take a sample from the embryo, from the part that will become the placenta, not the baby. So the baby is unaffected. And they test that to make sure, well, to do the testing in terms of the PGT-M testing for Ellis-Van Creveld and for the chromosome abnormalities. She then went on and told us that the embryos are stored and we'd get the results in about 2 weeks afterwards. So they freeze all of, so say if you get 5 embryos, they freeze all 5 once they've taken the sample, and then after 2 weeks they'll say kind of embryo 1 is affected, embryo 2 is a carrier, embryo 3 is not affected, embryo 4 whatever, and they kind of break it down that way. So they go ahead and freeze them at day 5 or 6, and then that's what we would implant later on if we get any that are okay. She asked about a sample from France after the abortion, so stored DNA in the French hospital, and we had no idea what she was talking about. Again, I think I mentioned this in the previous video because I'm filming this now like a month after the first consultation, or at least 2 or 3 weeks after, so we kind of got our things a little bit mixed up. So in the doctor's consultation, she didn't mention the French DNA, even though I mentioned that in the last video. It was actually during this consultation that she was like, yes, we'd like to request a DNA sample. And we were like, what do you mean? She was like, you know, from France. We were like, what's she on about, a DNA sample from France? And it took me a minute to click, and I was like, oh, from the abortion? She said, yeah. And I said, what, will they keep DNA? And she said, yeah, usually they do. And we must have signed papers saying it was okay, but just at the time we had no, I was just, you know, obviously in a mess. So I don't remember any papers that I signed. I mean, obviously signed lots of papers. I don't remember specifically what they said. So we had to then contact the French hospital and ask them if they had DNA. They have, so they've sent that, and that's now where we're at now. But yeah, that was a bit of a surprise for us. So if you have had a previous abortion and you're doing PGT-M testing, they might ask for a DNA sample from that abortion if they happen to have it. I didn't even know they would have that. I didn't know that was a thing. But apparently it is. And the reason that they want this is that it will improve accuracy. So obviously if you only have mine and Jeremy's chromosomes to look at, it might be hard to find the exact gene defect, whereas if you have a baby who has the gene defect and ours, it just adds to the kind of, just makes it more accurate basically, and it's easier to test for. So that's good that France has that. I just had no idea that they were going to have that. We then went through family history. So do any of our family members know that they are also carriers? Because obviously because I'm a carrier, one of my parents must be a carrier and they've passed it on to me. So either one of my parents or both of my parents are also carriers, which also means that maybe my brothers are carriers. So it was just interesting in terms of do any of them know that they're carriers? And if so, would they want to come forward and provide a sample as well that they could use to test to find, to get more accuracy and more detail? Unfortunately, none of our family have tested, so we can't use that. We don't really know where we got these genes from, if my mum or dad, and also if my brothers are also affected, or not affected, carriers. So that was interesting. So she asked us if we are related, which we've had before, because obviously it's a very, very rare disease. Syndrome, and they kind of want to make sure that we're not family members. And I was a bit like, no, I'm English, he's French, we met in Australia, we are not related, we don't know each other. And she said that over 80% of us are carriers of one condition, and if we expand our family with a relative, it will increase chance of that recessive condition. So I always thought that inbreeding, if you have babies with relatives, I know it's obviously bad, but I didn't really understand why. And it's actually just because 80% of people carry one syndrome, and if you're in the family, you're more likely to carry the same sort of gene defect, and that's why when you have inbreeding, babies often come out with different syndromes and abnormalities and things. So yeah, that's kind of why they asked us if we're related, and we are not, as far as we know. I mean, not for a couple of generations at least, a couple hundred years, I don't even know. We're not related. And then we talked about the minimum amount of embryos for testing if we don't have the DNA. So she was saying that if we have the DNA in France, if they have it, that could make it more accurate. They do have it. I can kind of tell you now, they do have the DNA, so that was been fine. But she said if they didn't have that, we would need to have a certain amount of embryos to be able to test. Now this was where I got a little bit because you can't guarantee how many embryos you're going to have. And she sort of explained to us that even if you have 10 embryos, but some of them get fertilized and then they stop growing at day 3, you can put that forward to be tested as part of the kind of testing for PGT-M testing for Ellis van Creveld. So it's not as if you need she said it could be 4 or 5. It's not as if you need 4 or 5 good embryos. It could be 4 or 5, including ones that died at an earlier stage. That's a little bit scary, so I'm glad we did have the DNA. so that we can hopefully not have to have a minimum amount of embryos. What else? We talked about needing to do blood samples at King's Fertility in London. So she said we need to speak to Apricity to get that organised and book in a blood test. That's coming up in one of the future vlogs. You'll see that. She also mentioned that we only have to do the PGTM workup once. So once we get it done this time, if we want to freeze embryos in the future, we won't have to do this workup section. We'll obviously have to pay for the testing, but not for the workup. So we could do this as many times as we want to and not have to do the workup every single time, which I think is quite good to know. So it's kind of just, it's one less thing to do next time, if there is a next time. Next thing she talked about was Cairo mapping, something like that, which is to figure out which part of the chromosome has the syndrome. So like I said, I was talking about kind of the rubber bands and which exact one it is. So she was explaining what that is to us. I'll pop a little definition on screen if I can find one. I asked her if you can choose gender, because I'd seen somebody on YouTube talking about embryo freezing or doing IVF, and they could choose whether they had a boy or a girl. And I was a bit like, oh my God, I would not want that responsibility. So I asked about that, and apparently it's illegal in England. Sex selection is legal, but the lab will know. But the main priority is just getting a healthy baby. So whether it's a boy or a girl, they don't mind. They're just going to choose the healthiest baby to implant and then go from there. And it'll be a surprise for us. So that was quite a relief. That was good to know. She talked about Mosaicism, I think it was called. I still don't really understand it. I think it was something to do with like, there's good cells and bad cells, and sometimes the results come back inconclusive or something. I'm not sure. I'll try and find a definition and pop that on screen as well. She said that PGT-M testing can have about a 98% accuracy, and if we do PGT-M testing, it's 98% accuracy when I'm pregnant, or if I'm pregnant when I'm pregnant in the future. We still have kind of questions about the baby and if they might still have Ellis Van Creveld. We can do an amniocentesis, or the other option, which I've forgotten what it was called and I didn't write it down. She said there's a 5% chance that the lab can't say with a high level of accuracy what the results is. And if that's the case, we can do a rebiopsy from grading of embryos. So I don't really know what that means. I've written it down and I can't quite remember. She said if we do implantation, don't have sex during that period, because obviously if I get pregnant with another egg whilst we're implanting with IVF, we won't know if the baby has the syndrome or not, basically. So that was kind of for future down the line. She said that there is a chance that there won't be any embryos that are affected or carriers and aren't chromosomally normal. And so I talked to her about that being my biggest fear, is that we'll go through all this, spend all this time, all this money, all this energy, and at the end have no embryos that we can use. Because say, for example, if you start off with 10 eggs and then you fertilise them and only 8 get fertilised, and then after day 5 or 6 you've only got 5 embryos left. From that 5, you might lose 25% to Ellis Van Creveld, a certain percentage to chromosome abnormalities, so you're only really left with maybe 1 or 2. And you might be left with 0, you might be left with 5. So I kind of talked to her about that being like my biggest fear, and she said, well, the best thing to do is to make a plan of action of like, okay, if we get this amount or less, what do we do? So we've kind of discussed it, and I think what we're going to do is either way wait until next summer, because I think if I have lots of embryos that are healthy, I might want to do more anyway just to have a backup. If I have like 2 embryos that are healthy, I might want to do it again, or if I have 0 embryos, I might want to do it again. And I think I'll either go one of 2 ways. I'll either be like, okay, we need to do it again next month, or fuck that shit, I'm not doing it again. It was too much hard work in terms of the injections, in terms of the money, in terms of the stress, all the rest of it. So I'll either go one of 2 ways. I'd be like, we need to do it again straight away, or never again. So we've kind of decided to give ourselves probably 6 months, 8 months to think it over. And in that time, I'll either be like, oh my God, I need to get it done, or I'll be like, no, I'm fine, never again. So it'll be interesting to see what happens. So we did decide we'll wait until next summer and just see how we feel, basically. But that was definitely worth talking about. I think it's, if you're thinking about doing embryo freezing, it is worth thinking about what you do if you don't get any at the end. So after talking to us about all the PGT-M testing, everything that's kind of, chromosomes, all the rest of it, family history, she said to us there's kind of different options. So option 1 is that we do PGT-M testing and transfer an embryo and get pregnant and do it that way. Obviously we can do that as well and do an amniocentesis as well, or we can conceive naturally and do an amniocentesis, or we can do IVF without testing, or we can do whatever and test after the birth if the baby has any features. So there's kind of different options in terms of what we do here. So obviously with this one we're going to do the PGT-M testing and then implant an unaffected and chromosomally normal baby. But there are, you know, we could still get pregnant naturally, we could still do another round of IVF and not test, there's kind of different things going on there. And we actually asked about amniocentesis, because when we did the amniocentesis when I was pregnant in 2017, so this was only 5 years ago, I think the percentage that they gave us, I remembered about 15%, I think Jeremy remembered about 30%. So between 15 to 30% chance of a miscarriage if you did amniocentesis. I remember thinking I didn't really want to do an amniocentesis, because if we did that and had a miscarriage but turns out the baby was fine, it's not worth the risk. But she explained it's actually about a 1% chance of miscarriage. She said that number has come down enormously over the past couple of years because they're just doing it so often. It's such a regular treatment, I guess, to do, not treatment, test to do. So it's only about 1% chance now of miscarriage. So that's kind of changed my views on that a little bit, because I was saying like I would never do it again, but because I thought the chance of miscarriage was like 30%, or yeah, just didn't seem worth it. But if 1%, maybe it is, I don't know. So she also said that if family members want to, they can go to the GP and ask for a carrier status report. So they can take my report in saying that my daughter or my sister or my whatever, cousin, whatever, has this carrier status of Ellis van Creveld. Can they also get tested? So my brothers, my family, my parents, my whatever can all go ahead and do that if they want to. No one has said they want to for now, which is fine. But that is something, if you're a carrier of something, your family can go ahead and test as well if they're curious. So the next steps was we needed to get in contact with France and see if they had the DNA, and then also book in ourselves to get the blood work done. So again, you'll be seeing those vlogs coming up. So the blood work takes about 4 to 6 weeks to get going, to do it. And then the last thing that she kind of said was that at my age, because I'm 34, we have a 40.5% chance of a chromosome abnormalities. And she said that because of previous miscarriages, don't be alarmed if there are some embryos with chromosome abnormalities. For example, embryo 1 has a problem with chromosome 2, and then embryo 2 has a chromosome abnormality with chromosome 17. So it's not necessarily going to be the same chromosome abnormality for each embryo that does have an abnormality, but just that it might be quite high. Now this got me quite upset, because if there's a 40% chance of a chromosome abnormality plus a 25% chance of an EVC affected baby, that means 65% of our embryos are gone. Before we've even started, they've gone. So you've then got to hope and pray that at least 1 or 2 or 3, however many we can get, are, you know, they fertilise, they stay alive to day 5 or 6 to be able to get tested, and then that they are unaffected. So we really, honestly, we've lost, like I said, 65% of our embryos before we've even started, which is pretty shit to find out. And I had a bit of a cry after that, and I was like, what the fuck's the point? Why are we doing this? It's a waste of money, waste of time. It's not going to result in anything. And I just felt really defeated and deflated that we're going to lose 65% before we even start. It just seems like such a huge number to be battling against. And we actually asked about the chromosome abnormalities, because we weren't sure if a baby has a chromosome abnormalities but you implant it anyway. And she said, to be honest, they wouldn't implant it, but most of the time with miscarriages, it's a chromosome abnormality. And she said that even if they are born, they have pretty severe difficulty. So she said obviously there is Down syndrome, but there's also other ones where the baby dies quite soon, is in lots of pain, goes to hospital a lot and dies within, you know, a year or 2. So it's just like, that's not something I want to go through. I don't think it's something that anyone wants to go through. So she said, you know, it's not as if you could have a chromosomally abnormal embryo and implant it anyway, because it probably wouldn't result in a live birth. And if it did, it wouldn't be good, basically. Her words, not mine. I know it's always really hard when you talk about things with Down syndrome, and it's such a weird minefield, but they wouldn't even implant it. It's not even our choice. And then the last thing was, oh yeah, that I cried afterwards because of the numbers, and then to contact France and organise the blood test. So that was kind of, I know that was a lot of talking. I hope, how long have we been filming for? Half an hour. So I hope, half an hour? Blimey. So I hope there was some information in there that you found useful. If you have any questions, please do let me know in the comment section down below. I'm very happy to answer any questions, or you can message me on Instagram otherwise. But yeah, that was like, I just wanted to share this information, partly for me to be able to look back on, also for the people who might be going through PGT-M testing or IVF or any of this. It's an absolute minefield. It's just so hard to know what's going on and what to expect. So if you happen to be with Apricity, if you happen to then go with King's and do PGT-M testing, this is the sort of conversation that you will be having, talking about family history, talking about what chromosomes are and cells are and your specific disease that you're a carrier of or the syndrome you're a carrier of. You might also be talking about percentages and the actual process, what happens, carrier mapping, mosaicism, all these different things are things that you're going to probably be talked to about. So yeah, I hope this was somewhat helpful. I'm sorry this video was so long. I tried to be quick and it was still half an hour. But thank you for watching. If you enjoyed this video, make sure you subscribe. We have vlogs coming up very soon. Like I said, there's the one where we go to the doctors and do an ultrasound and do a blood test. There's also another one where we kind of give a life update because the pricing changed and lots of different things went on. So that video is coming very soon as well. So make sure you subscribe. I hope to see you soon and thank you for watching. Bye.